while I'm introducing today. So today's speaker is Danielle Harper, who's a PhD student in my lab. So it's a real pleasure to get to introduce her. So Danielle is a graduate of Trent University, and she had the misfortune of joining my lab in September of 2020. And I guess everyone will remember what was going on at that point in time. So she kind of got stuck at home, and she accomplished something in her first semester that nobody else has done in the pathology graduate program in as many years as I've been around. She finished all of her coursework for the PhD program in the first semester. So that's a bar that I think it'll be difficult for anybody to match after that. But I guess Danielle was very ambitious and didn't have much to do because of the pandemic. So she tucked into all the coursework, and of course things did get going in the fall, so she was able to complete the last course that she had to do then. So Danielle's been with us now for, this is her third year. And so she gave her first talk in the department in the winter of 2021. So we're about 2 years past that point now. So we get the pleasure of having her come back and tell us how things have been going. So she's accomplished great things in the last 2.5 years. And she also accompanied the Calpain part of the lab to Lisbon, Portugal last summer. And Danny actually gave us matching t-shirts or sweatshirts to wear, and I should be wearing mine today. I still make use of that, but that's an example of her lab spirit. So Danny, you have the floor. Take it away.
Perfect. Thank you, Peter. You can hear me okay still?
Oh, yeah.
Okay, perfect. So thanks everyone for joining on Zoom today. I'm sorry we couldn't be in person. I'm just getting over COVID again, so just wanted to play it safe. So the title of my talk today is Breaking Down the Role of Calpain Proteases in Triple-Negative Breast Cancer Tumorigenesis, Metastasis, and Drug Sensitivity. So before I jump in too much, I just wanted to start with a brief overview of breast cancer epidemiology. And as you can see by the pink areas on the map here, breast cancer is the most diagnosed cancer in females around the world, and it accounts for about 1/4 of cancer diagnoses in women and is the second leading cause of cancer-related deaths in females. It's estimated that 1 in 8 Canadian women will be diagnosed with breast cancer in their lifetime. And fortunately, breast cancer mortality has been on the decline since the 1980s. And this is largely due to early detection through mammography screening programs. However, a recent study published in 2021 actually revealed that breast cancer mortality rates have stopped declining in women younger than 40 years old. And what's more is that breast cancer incidence in that population of women less than 40 is increasing, and younger patients are more likely to be diagnosed with more aggressive tumors. So several factors are used to guide breast cancer treatment and prognosis in clinical settings. And the two that I'd like to highlight are clinical stage and molecular subtype. And I'm sure everyone's familiar with the idea of staging. So the idea that stage 1 tumors are localized to the primary tumor. Stage 2 and 3 may see some regional metastasis into surrounding tissues, and then stage 4 is marked by distant metastasis to the brain, lungs, liver, bone, for example. So there are 4 main breast cancer subtypes, and these are characterized by the presence or absence of estrogen and progesterone hormone receptors, so ER and PR, as well as the HER2 human epidermal growth factor receptor. And in addition to early detection, improved survival can be attributed in part to the development of targeted therapies that take advantage of some of these molecular characteristics. So just as an example, there's the estrogen receptor modulating drug tamoxifen, and this can be used for ER positive tumors. And then there is the, going the wrong way. The monoclonal antibody trastuzumab that is used for the treatment of HER2 positive tumors. And as you can see in this table here, which represents survival rates across the various stages. Survival does decrease as stage increases, but we see the worst outcomes in triple-negative breast cancer. And as the name suggests, triple-negative breast cancer, or TNBC, is characterized by a lack of estrogen, progesterone, or HER2. And it's considered to be the most aggressive breast cancer subtype. It accounts for only about 15% of all cases, but it's responsible for about 25% of breast cancer-related deaths. So furthermore, triple-negative breast cancer disproportionately affects young women as well as young Black women, and racial disparities can continue to contribute to poor outcomes in this population. So overall, triple-negative breast cancer is traditionally associated by this lack of therapeutic targets, but this really is an oversimplification of a highly heterogeneous subtype that hasn't yet been fully characterized. So researchers have started to explore some of the unique characteristics of triple-negative tumors in order to further classify them into additional subtypes. And right now, the most widely accepted classification system recognizes 4 TNBC subtypes as outlined here. So there are those that have defects in DNA repair mechanisms, those with high expression of the androgen receptor, those with dysregulation in pathways involved in growth and dysregulation, and then tumors with high levels of immune infiltration. So sort of by looking beyond the cell surface, sort of both figuratively and literally, we're learning more about what drives these triple-negative tumors and some of the therapeutic approaches that we can take to improve patient survival. And I just wanted to take this opportunity to introduce this idea of synthetic lethality. And this is the idea that a mutation in 2 genes simultaneously will result in cell death when a mutation in either of those genes individually does not. So this concept of synthetic lethality, we can take advantage of it from a therapeutic perspective to target either 2 genes or 2 pathways simultaneously that will result in cell death. And I just wanted to give a quick example while I'm on this slide, and that's of PARP inhibitors, which are so a relatively new treatment for triple-negative breast cancer. So PARP inhibitors inhibit the poly-ADP ribose protein, which is responsible for single-stranded DNA repair. And essentially, when you block PARP, you get an accumulation of these single-stranded breaks that turn into double-stranded breaks, which then require the protein BRCA or BRCA to fix these double-stranded breaks. So approximately 10% of triple-negative breast cancer patients present with mutations in BRCA1 or 2. So when they lack this BRCA protein, blocking PARP results in this synthetic lethal form of cell death. So despite these emerging treatment options for TNBC, there are still high rates of recurrence, metastasis, and drug resistance among these patients. So this really emphasizes a need for a greater understanding of TNBC biology, as well as the identification of novel therapeutic targets. And this brings me into CalPANES. So I'll just start by introducing the purpose of my project, which is to explore the role of calpain proteases in breast cancer tumorigenesis and metastasis, and identify specific therapeutic agents, specific drugs that will synergize with calpain disruption in models of triple-negative breast cancer. So I've just sort of included this synthetic lethality model just to highlight this idea that I'm looking to identify drugs that can be combined with calpain inhibition to block some kind of protein involved in the tumorigenic process in order to result in cell death. So now I'll just go over some background on calpains. So calpains are a family of 15 calcium-activated cysteine proteases, and there are 5 main classes of human proteases, and they're outlined in this figure here. And they are classified based on their reaction mechanism. So as the name suggests, cysteine proteases, they contain a cysteine residue that acts as the nucleophile within their active site, and this catalyzes the hydrolysis of peptide bonds. So calpains regulate the function of their substrates through proteolytic processing, so through these hydrolysis reactions. And there are other mechanisms within the cell that lead to proteolytic processing. So of course, we have these different types of proteases here. There's also the proteasome complex or lysosomes that lead to degradation of substrates, many of which are ubiquinated. However, one of the unique things about calpains is that their cleavage doesn't necessarily degrade the substrate. It can change the function of the substrate through these cleavage events. So there are still a lot of questions regarding substrate specificity for calpains, but overall, substrate recognition is thought to depend on favorable binding interactions that incorporate specific amino acid preferences. So an example is the presence of a pest motif and other things like secondary structure as well. So my project focuses on calpain 1 and 2. So here is just an overview of where various calpain isoforms are expressed within the human body. So calpain 1 and 2 are ubiquitously expressed, and they are heterodimers consisting of a large catalytic subunit. And this subunit is encoded by the CAPN1 gene in calpain 1 or the CAPN2 gene in calpain 2. And both of these isoforms share a common regulatory subunit, which is encoded by the CAPNS1 gene. These subunits interact through their PEF domains, and the small subunit is required for stabilization of the large subunit. So notably, genetic disruption of CAPNS1, the small subunit, leads to a loss of both calpain 1 and calpain 2 activity. And this is something that'll come up when I talk about my different models. So like I mentioned, calpain is a calcium activated protease. So its activity depends on the proper orientation of its catalytic triad within its active site. So within calpain 1, for example, the catalytic triad consists of cysteine 115, histidine 272, and asparagine 296. And it's this calcium binding that leads to the proper orientation of the enzyme and causes this catalytic triad to come together. So if I turn off my laser pointer, I think I can play this animation. This comes from Peter Davies' lab here at Queen's, and this just shows calcium binding leading to that conformational change. So here there's a total of 10 calcium ions binding in total. So calcium binding down here at the penta-EF hands triggers the release of the anchor helix and rearrangement of the large and small subunits. And then binding in these protease core domains, so PC1 and PC2, this is what causes the formation of the catalytic triad and activation of the enzyme. So calpains are activated by calcium, but they are regulated by their endogenous inhibitor, calpistatin. And in this figure here, it is this purple peptide. And it forms 3 alpha helices when it's in complex with calpain. The A and C peptides stabilize the interaction between calpistatin and calpain, and then the B peptide actually blocks that active site. And here I'll just mention that there are currently no isoform-specific pharmacologic inhibitors of calpain that are commercially available. So you'll see this is why we're using genetic models in order to inhibit calpain. There are some commercially available inhibitors that are marketed as calpain inhibitors, but most are sort of general cysteine protease or general protease inhibitors that lack specificity. And there is ongoing research to identify and validate potential small molecule inhibitors that could be adapted for clinical use eventually. So calpain dysregulation has been associated with a range of physiological conditions, and we call these calpainopathies. And some of these conditions have been linked to specific mutations. So for example, mutations in CAPN5 have been linked to autoimmune uveitis, retinitis, and other retinopathies that lead to blindness. Similarly, calpain 3 null mutations are associated with limb-girdle muscular dystrophy. And then calpain 1 mutations have been linked to cerebellar ataxia, which manifests as difficulty with voluntary movements, including walking and fine motor skills and things like that. So there are other calpainopathies that are rooted in post-transcriptional dysregulation. So for example, calpain 2 dysregulation is thought to contribute to a neuropathogenesis of Alzheimer's disease. And interestingly, calpain 2 is widely thought to be neurodegenerative, while calpain 1 is thought to have a neuroprotective effect. So for the remainder of the presentation, I'll be focusing on calpain's role in cancer and our hypothesis that targeting calpain 1 and 2 in breast cancer will enhance the efficacy of anti-cancer drugs. So I just wanted to provide some clinical evidence in support of this hypothesis. So translational studies have reported a link between calpain expression and clinical outcomes. So both of these studies come from Dr. Stuart Martin and Dr. Sarah Storr's group in the UK. In one study, they reported that high calpain-1 expression was linked to trastuzumab resistance and poor survival in HER2-positive breast cancer. And in another study, they found that high calpain-2 expression was linked to poor clinical outcomes in triple-negative breast cancer. And I just wanted to highlight a few calpain substrates that are implicated in cancer-related signaling pathways. And these include pathways with effects on cell survival, cell cycle progression, and apoptosis. So these are just a handful of calpain cleavage events that may contribute to cancer cell signaling. And a little bit later in the presentation, I'll take a closer look at the PI3 kinase AKT signaling pathway. So with that, I just wanted to review my research aims, and I'm going to spend the majority of the presentation focused on these first two, being identifying synthetic lethal interactions between genetic calpain disruption and clinically approved drugs, and then starting to explore some calpain substrates that are involved in signaling pathways that are contributing to tumorigenesis and metastasis. And then I will come back to sort of this future aim a little bit later in the presentation. So my first aim is to identify drugs that will be more cytotoxic to calpain deficient cells. So I just wanted to review some previous findings from our lab that support this hypothesis. So this first study was conducted using primary mouse embryonic fibroblasts, or MEF cells. So these are not cancer cells, but they were, so we have wild type and CAPNS1 knockout, so small subunit knockout MEF cells. So these lack both calpain-1 and calpain-2 activity, exposed to a variety of stimuli, as we can see here. And calpain deficient MEFs were more resistant to some treatments, including serum starvation, UV exposure, or the chemotherapeutic drug etoposide. But conversely, they were more sensitive to treatment with TNF-alpha or staurosporine. And these Western blots on the right here just show PARP cleavage as a way of looking for a marker of apoptosis. And we can see that when treated with etoposide, we see greater levels of PARP cleavage in the wild type cells, whereas alternatively, when cells are treated with staurosporine, we see greater levels of PARP cleavage in the CAPNS1 knockout cells. So this, again, is just an example from a non-cancerous cell line that illustrates that calpain's role in apoptosis is likely context dependent and may differ based on the circumstances and the stimulus that the cells are treated with. So more recently, our lab demonstrated that shRNA-mediated knockdown of the small subunit was associated with increased sensitivity to 17-AAG, which is an inhibitor of the molecular chaperone HSP90. So here we can see an increase in cell death in the small subunit knockout cells. And then in tumor engraftment models, there's a reduction in tumor growth when this shRNA knockdown is combined with 17-AAG treatment, as well as a reduction in the number of lung metastases. And the relationship between HSP90 and calpain isn't fully understood at this point, but there was rationale to target both based on some overlap in calpain substrates and HSP90 clients. And then similarly here in a HER2 positive mouse model, calpain knockout cells were found to be more sensitive to the receptor tyrosine kinase inhibitor lapatinib, as well as the anthracycline doxorubicin. So you can see that here as well. All right, so I've mentioned my aim. I just wanted to briefly review my workflow for this aim. So for this part of my project, I'm using the MDA MB231 human triple-negative breast cancer cell line, and I've engineered an shRNA knockdown cell, small subunit knockdown cell line, and I'm performing a medium throughput screen of the target mole drug repurposing library in collaboration with Dr. Franco Visukumar's lab at the University of Saskatchewan. So the target mole drug repurposing library is quite similar if you're familiar with the Prestwick library. It's about 2,000 FDA approved drugs. And the idea of using this drug repurposing approach is sort of to, rather than having to develop novel drugs that may work synergistically with calpain deficiency, trying to identify drugs that are already in clinical practice that can be repurposed for use with a potential future calpain inhibitor. So the way that the screen is being performed is with the InQSight live cell analysis system. And we do have one of those available to us here at Queen's. However, Dr. Vijaykumar's lab has access to a liquid handling robotics facility where they're able to carry out this higher throughput type of screening. And essentially, small subunit knockout and control cells are being exposed to various concentrations of the drugs within the target mold library, and then cell confluence is being measured over 5 days. And then I'll show on the next slide what some of these preliminary results are looking like. But once we have some hits from this initial medium throughput screen, I'll be moving on to in vitro validation here with our own InQSight, as well as some other assays, including Presto Blue. And then once we have some more validated hits, we can move on to mouse mammary fat pad engraftment models. Yeah, so I'll just highlight some results sort of just starting off with. So these are my the immunoblot from my cell line. I just wanted to show here my knockdown cells alongside a CRISPR-Cas9 knockout, and just show that the knockdown is comparable to the CRISPR knockout. And then here is a casein zymogram, which is used to assess calpain activity. And we see a loss of both calpain-1 and calpain-2 bands in the Cas9 knockdown. These sort of scatter plots just show some of the preliminary results from the target mold drug screen. And the idea here is that the results are presented sort of as a standardized fold change as the confluence has been measured over these various time points. So the drug hits that we are interested in are those that we see little to no change in the control cells, but we see a reduction in that in cell proliferation over time in the knockdown cells. And then here I just wanted to highlight a couple of the hits that came out of our of the first replicate of this screen, just to show what sort of these confluence curves look like over time. So here we have Wabane octahydrate and then also gefitinib. So moving forward, I'm starting to pick out some of the initial hits that we've been presented with, validating those in vitro, and then eventually moving in vitro, and then starting to look at what are some of the drug targets of these hits, and maybe starting to piece together a story about how calpain may be regulating sensitivity to these specific drugs. All right, so before I move on to the next aim, I wanted to just briefly review calpain's role in cell migration. By highlighting some substrates involved in the formation of invatopodia and lamellipodia. So calpain cleaves several substrates to contribute to cell invasion, including PDB1B, SARC, and cortactin. And then calpain cleavage also helps with focal adhesion turnover, which is essential for lamellipodia formation and cell migration. And these include proteins such as focal adhesion kinase, talin, vinculin, and paxillin. So my second aim is to identify calpain substrates that are involved in signaling pathways that contribute to tumor genesis, metastasis, and drug sensitivity. And to do this, I'm moving into another model system, and these are AC2M2 cells, which were derived in the Elliott lab. And essentially, for this part of my project, I'm characterizing the phenotypes associated with loss of calpain in these cells, and then starting to look a bit at the PI3 kinase AKT signaling pathway and how that pathway is how that pathway is dysregulated by a loss of calpain. So like I mentioned, the AC2M2 cell line is a mouse triple negative-like cell line. It was derived from this original cell line called SP1. That was passaged 3 times through the lungs to create this highly metastatic variant. And what's unique about the AC2M2 cell line is that it has this autocrine HGF loop. So HGF is secreted by these cells, and HGF binds to the c-Met receptor, which activates downstream signaling, including the AKT pathway. So that's just one thing that's unique about these cells. for these cells, I used CRISPR-Cas9 knockout to establish CAPN-S1, CAPN1, and CAPN2 knockouts. And here I'll just highlight again that a loss of CAPN-S1 is also associated with a loss of CAPN1 and CAPN2 large subunits. And again, here is just the zymogram to show that loss of both CALPIN1 and CALPIN2. So I'll just start by going through some of the phenotypes observed in these cell lines, starting with migration. So this was a single cell migration experiment where cells are seeded quite sparsely, and then using the IncuCyte, their trajectory over time is tracked, and it produces these nice spider graphs where each line corresponds to the trajectory of an individual cell. And CAPN-S1 knockout was associated with a significant reduction in the average speed of the cells, as well as a significant reduction in their total displacement, and a slight reduction in their directionality, which just refers to their persistence in one direction. The next assay that I wanted to show was the DQ collagen assay for collagenase activity. Also used as an assay for cell invasion. So in this assay, wells are coated with collagen that is conjugated to an excessive number of fluorescent dye molecules. And in this case, it's a fluorescein conjugate that fluoresces green, as you can see here. So in the absence of cells, the signal quenches itself because there are so many dye molecules present. But as the cells begin to invade, they release matrix metalloproteinases, which results in hydrolysis of this collagen ECM. And this causes separation of the dye molecules, and then you start to see that fluorescent signal. So to quantify the results of this assay, I divided the total area of green signal by the number of nuclei in the field of view. And interestingly, these results seem to suggest that CALPIN2 may play a greater role in cell when it comes to cell invasion, as we see a decrease in that DQ collagen signal in both the small subunit knockout and the CALPIN2 knockout. All right, and then moving on to some in vivo assays. So CAPN-S1 knockout AC2M2 cells have reduced ability to colonize the lungs in a tail vein model of metastasis. So in this assay, 1 million cells are injected into the tail vein of mice, and then I look at the lungs at these various time points. And there's a reduction in the number of metastatic lesions at 24 hours and 96 hours, and then a significant reduction in the metastatic burden or the total area of lung occupied by lung mets at 12 days. Similarly, in a resectin weight model of metastasis, CAPN-S1 knockout was associated with a significant reduction in lung metastasis. This is 12 days post-resection. That the lungs were harvested and imaged. And these are just some preliminary images looking at also the CAPN1 and CAPN2 knockouts. And I'm currently working to validate these phenotypes and get more replicates. But the preliminary results are promising as we see a reduction in the small subunit in CAPN1 knockout and CAPN2 knockout 12 days post-resection. And I'll just highlight here too, this pale green is just background signal. So the actual mets themselves are these very bright areas. All right, so I mentioned some of those substrates that are involved in migration and invasion. So I've looked at those in vitro. This first blot is looking at cortactin cleavage, and these are on cells that were treated with various concentrations of calcium in addition to calcium ionophore in vitro. So it was the actual cells in culture that were treated, not the lysates. And here we, in the CAPN-S1 knockout AC2M2s, not only do we see an increase in the total amount of full-length cortactin, we don't see this cleavage product that we see in the wild-type cells at 1 and 5 millimolar concentrations of calcium. And then similarly, it doesn't seem to be a calcium concentration, there doesn't seem to be a dose response here, but we do see focal adhesion kinase cleavage products in the wild type cells that we don't see in the CAPN-S1 knockouts as well. So I just wanted to highlight focal adhesion kinase here, a member of the focal adhesion complex, and then cortactin, which again plays a role in lamellipodia and invatopodia formation. And then getting into some of the signaling, I just wanted to talk about the AKT pathway. So AKT is one of the most frequently upregulated proteins in cancer. And briefly, its activation requires phosphorylation at these two residues, so threonine 308 by PDK1 and serine 473 by the mTOR complex 2. And AKT promotes cell survival through several mechanisms, but I just wanted to highlight one here, and that's through inactivation of the transcription factor FOXO3A. So when AKT phosphorylates FOXO3A, it leads to its relocalization from the nucleus into the cytosol where it gets degraded. And yeah, so FOXO would normally promote activation of P27-KIP1, which prevents cell survival. So there are some calpain substrates implicated in this pathway, including PTEN, PP2A, and FLIP. So these have been identified as calpain substrates, but not necessarily in a cancer context. In the case of PTEN and FLIP, this observation has been made in neuronal cells. So at this point, it's not clear if calpain may be regulating this pathway through effects on these phosphatases, but that's something that I am interested in. And this is based on a previous observation by a postdoc in our lab, who saw that when calpain-2 was knocked down, there was an 80% decrease in AKT phosphorylation at serine 473, as well as an increase in total levels of PP2A and P27-KIP1. And it was also observed that calpain knockdown led to an increase in nuclear localization of FOXO3A, as we can see here, compared to the wild type cells. Another interesting observation from Waichi, who was the postdoc, that has not been published, is this idea that AKT activation could be induced by treating with the PI3 kinase inhibitor degulin in wild type cells, but not in calpain-2 knockdown cells. So there's this idea that loss of calpain is associated with a decrease in AKT activation. So this is something that I've started to explore. So when treating with degulin, I haven't seen an indication that I've been able to prevent activation of AKT phosphorylation, but I have seen a reduction in total levels of phosphorylated AKT overall. So here the wild type compared to the small subunit knockout, as you can see in this plot here. However, when cells were treated with rapamycin, which is an mTOR inhibitor, I have been able to prevent this activation of AKT, as shown by phosphorylation at serine 473. One other thing that I've noticed, and from this plot, it's not entirely evident as there is a bit of a loading issue here, but I noticed an increase in total levels of PP2A. So I wanted to go and see if there was a difference in PP2A cleavage between wild type and small subunit knockout cells. And here we can see, I don't know how well you can see it on your screen, but there's a 30 kilodalton cleavage product in the wild type cells that I've not seen in the CAPN-S1 knockouts. So there's still lots to be done in terms of understanding how calpain might be regulating AKT activation in AC2M2 cells. But I'm continuing to explore the involvement of these three phosphatases, in addition to the c-Met receptor, which is an upstream regulator of this pathway. And of course, we have this autocrine HGF c-Met loop in the AC2M2 cells. So I'm really excited about these observations and looking forward to hopefully sharing an updated version of this story and trying to piece together this a little bit more. So I'll just go on to talk about that third aim there, which is sort of a relatively new aim and sort of somewhere that I want to take this project. And this is the idea of exploring how calpain disruption in triple-negative breast cancer cells and in hematopoietic cells might affect cancer immune responses. And I'll come back to these hypotheses in a second. I just wanted to review sort of some rationale for exploring calpain's role in anti-cancer immune responses. So this sort of goes one step further from exploring calpain's cancer cell intrinsic role to roles in the immune system and how we might be able to take advantage of that therapeutically. So there are several established roles for calpain in immune cells. but very few in a sort of cancer context. Calpains have been shown to promote macrophage apoptosis, neutrophil necrosis, and lymphocyte proliferation, in addition to T cell activation. They've also been implicated in cytokine release and activation of NF-kappa B by cleaving I-kappa B. There's one really interesting study from 2021, which showed potential anti-tumor effects associated with blocking calpain, suggesting that calpain-mediated cleavage of Beclin-1 is responsible for recycling PD-L1 to the tumor cell membrane. So this is a unique observation that may also provide some rationale for targeting calpains. So if I come back to my hypotheses, I'm interested in looking at tumor growth kinetics and metastatic burden in immune competent mice versus immune deficient mice, because all of my work to this point has been done in RAG2s. And then I also hypothesize that systemic calpain inhibition may have off-target effects on immune cell function that could either enhance or hinder anti-cancer immune responses. And this kind of comes back to this idea that moving past our genetic models, when a pharmacologic calpain inhibitor is developed, we need to ensure that there are not off-target effects on immune cells, or if there are, we need to know what kind of effect that will have on anti-cancer immune responses. So my sort of proposed experiments for this aim, as I mentioned, first is to observe differences in tumor growth and metastasis in immune competent and immune compromised mice. So to do this, I'll be using the EO771 cell line, which is a triple negative cell line that is derived from the BLAC6 mice. So this is the syngeneic model to observe differences in tumor growth and metastasis between these mice with intact immune systems and the immune compromised mice. Then I'm also interested in establishing a mouse cell line that lacks calpain in its hematopoietic cells. So to do this, we're hoping to cross our CAPNS1 floxed mice with mice expressing a VAV-CRE transgene, so to hopefully knock out calpain in those hematopoietic cells. And from there, we can do immunophenotyping of the spleen to begin with, and then perform some orthotopic memory fat pad engraftments and start to look at things like immune infiltration into the primary tumor, as well as the lungs and the spleen. So under those circumstances. So overall, I'm looking to see if calpain inhibition might synergize with existing therapies to potentially offer a new approach, a new combinatory approach for the treatment of aggressive triple-negative breast cancer, and hoping that a greater understanding of calpain's role in breast cancer tumorigenesis and metastasis might guide new applications for pharmacologic calpain inhibitors when they are developed. And it's important to explore both cancer cell and immune cell intrinsic roles for calpain, to have a greater understanding of potential off-target effects of calpain inhibitors in cancer therapy, and hopefully be able to apply calpain inhibitors in combination with drugs that are already clinically available. And with that, I'd just like to thank all of the members of the Greer Lab, especially Dr. Greer, and our collaborators at the University of Saskatchewan as well, as we're performing this medium throughput screen. And I'm happy to take any questions.
Great. Thank you very much, Natasha. Or Natasha, I'm looking at Natasha on your slide. All right, so thanks, Danny. And I'll invite questions from the audience at this point. So you can use your hands up function, or you can just turn on your mic and chime in if you like. While people are thinking up some interesting questions. One thing, Danny, that occurred to me when you're giving the analysis of AKT is, have you looked at 308, 3-ene-308 in your immunoblotting analysis yet?
I've looked at it once and I didn't see any difference, but it's definitely something that I'm going to continue to keep probing for.
Because yeah, most of the time we look at 473, but I think that Wai-Chi did some analysis of 308, but I'll have to go and dig it up and see what it was.
Yeah, especially if this effect is mediated by calpain cleavage of PP2A, we may see differences in 308 as well.
Yeah, so another thing that is an obvious experiment that I don't know if we've talked about doing, but looking at PDL1 expression,
Yep.
steady state levels in your various panels of cells now is probably a good place to start with that Becklin idea.
Yeah.
And my last question is about the EO771 model. So remind us where you're at with EO771s, or because that's what you're hoping to do in engraftments and genetic engraftments with.
Haven't started that yet. We're currently working on doing the small subunit knockdowns in both EO771s, and then we have a metastatic variant, so the L3 cells. So doing small subunit knockdown in both of those.
Okay.
Yeah.
Okay, so that's what you're doing with Samantha, right? Okay, so questions? Danny's here until 5 o'clock. Or not. Dr. Lillicrap, go ahead.
Yeah, thanks, Peter. Daniel, can you hear me okay?
Yep, yep.
Yeah. So thank you for a very nice seminar. Lots of interesting stuff. And I apologize if my question is just missing things that you've said and I've misunderstood you. So the question really relates to, I mean, you're using these strategies to look at triple negative breast cancer, but I didn't hear you talk a lot about cell type specificity or even organ specificity. I mean, calpains presumably are widely expressed.
Yeah.
Their substrate specificity is fairly promiscuous, and so some of the things you described early on, for example, where you talked about capin S1 knockdown and chaperone inhibition, HPS 90, will kill any cell, I suspect. I mean, it's not going to do, cells are not going to do well with those combinations. So could you make some comments about the connection to breast cancer and triple negative breast cancer, particularly around the calpain knockdown or combinatorial methodology?
Yeah. So I guess I'll start by saying, yes, so calpain 1 and 2 are ubiquitously expressed, so they're expressed throughout the body. There is some evidence based on immunohistochemistry that calpain 1 and 2 are overexpressed in breast cancer tissue compared to normal tissue. So the thought is that there may be upregulation of calpains in the cancer that could be potentially driving the cancer or driving metastasis. The one thing about those, so these studies, for example, where we're looking at calpain expression and its correlation with survival, this is just based on IHC staining, so we don't know much about the activity level of calpain in those tissues. And that's one of the challenges, is not knowing necessarily the level of activation in these tissues. And I'm not too familiar with any studies that have looked specifically at calpain activity within tumor samples versus like normal tissue controls.
Thank you.
I see a comment from Yvonne. Since you see an anti-metastatic effect of calpain knockout in both engraftment and in the tail vein models, what's your best guess at this point for at what point and how is metastasis abrogated on a cellular level?
So I think the results so far suggest that there may be roles at different levels of the metastatic process for calpain and cell survival. So it could be in part due to, so tail vein, it could be sort of an anti-anoicus or anti-cell death within the bloodstream effect, or it could be some effect of the cells being able to proliferate once they've actually seeded in the lungs, versus in an engraftment model, it could be due to the extravasation of cells from the primary tumor into the bloodstream, and then from there, their ability to survive and seed in the lungs. So I think there are definitely potential for multiple points of I guess, multiple points in the metastatic process that calpain could be playing a role.
So I think Dr. Lillycraft's question probably, if I understood it correctly, relates to this concern that inhibition of something that's so widely expressed might be toxic, especially in combination with other therapeutics. So what can you tell us about what people have seen when they've knocked out calpain 1 and 2 by using the small subunit knockout mice? What they've seen with phenotypes in those animals where they've essentially abolished calpain 1 and 2 activity in any particular lineages?
Yeah, so small subunit knockout is embryonic lethal. Sorry, what was the second part of that, Peter?
Well, that's during embryogenesis, but when you so if you're doing a complete knockout, yeah, you're going to get embryonic lethality. But if you knock it out in specific lineages, not everywhere, what do you see?
Like in specific
Yes.
types.
So what happens when you knock it out in neuronal cells?
Oh, so well, you're going to get ataxia with a calpain 1 knockout. So that's sort of.
I don't know if ataxia was ever characterized in those early studies, but the mice were fine. They did have some LTP, long-term potentiation defects, which is seen in a lot of knockouts. But I think the good thing is that you seem to be able to knock out calpain 1 and 2 in adult animals in various tissues, and they don't seem to be too seriously affected.
Yeah.
At least, and of course that isn't what you'd be doing therapeutically. You'd be inhibiting it episodically, not completely forever. Yeah. David, did you have another question, or did you just have?
Well, I did, because it seems that everyone else is being shy, so I don't want to.
Yeah, go ahead.
So your synthetic lethality proposal.
Yeah.
So I wonder whether you can just sort of tell us where you think these two hits may be. So you've got calpain knockdown on one hand, which could be in a whole bunch of different places, I guess. And then you've got the screen you're doing where you're beginning to get some hits. What do you think are the so you have a couple of graphs here at the bottom of this slide that maybe you could spend a bit of time telling us what you found there on this very early information. What are the more likely double hits that will be synthetically lethal?
I mean, I think sort of in general, hopefully it'll be something where its drug target is somehow related to calpain signaling in terms of some kind of substrate. What's interesting about this one hit Wabane is it's a sodium potassium pump inhibitor, and it actually increases intracellular calcium levels. So there could be some kind of mechanistic link there between a pro-survival function for calpain being activated when treated with this drug versus calpain deficient cells. So that's sort of just speculative at this point, but it's sort of one link to make between calpain and the mechanism of action of this drug, whereas gefitinib is an EGFR inhibitor. And not in this model, but in another model, the mammary tumor epithelial cell line that we have, which is a HER2 positive model, calpain knockout cells were more sensitive to lapatinib, which I showed on the previous slide. So there's sort of that mechanistic link there between potentially EGFR signaling. And those cells also had differentially phosphorylated EGFR as well. So there could be something potentially going on there.
Thank you.
Okay. If there are no more questions, please join me in thanking Danielle for a wonderful talk. And I'd remind you all that next week we have I think this is, who's up next week? That's probably Iman. I think Iman Radwan is giving a talk next week. So we'll see you all again next Tuesday at 4 o'clock.